Hello, I'm Yegor Prikazuk, and this is the user guide of SCOPE, Soil Canopy Observation of Photochemistry and Energy Fluxes model. Today, we are going to talk about the major application, solar-reduced chlorophyll fluorescence. What I expect you have already done is you went to our GitHub and you downloaded the model with download zip. After that, you have unzipped the file, and now you can navigate to Scope Master, and you see these files. Also, of course, you have MATLAB, which would normally start with double click of Scope. See that the Scope is open, and we run it for the first time. The first run has 100 simulations, and hopefully you get the same message that the output is the same as in the verification dataset, which means we are good to go. What I also expect you to do is to find our documentation. The link is directly in the README. In the documentation, this application is listed under model capabilities, fluorescence. There are two very important caveats. One is that we are talking about something, the signal, the light that is emitted inside the plant leaf by some of photosystems. Some of them emitted. And this is typically measured with help of another technique, with help of active fluorescence, with help of PAM, pulse amplitude modulation. But in SCOPE model, we are talking about passive solar-induced fluorescence. So solar-induced, it can only be passive because it's induced by the sun. It's in steady state. There are applications of SCOPE model that are also possible with PAM data, but the spectra that we'll see and we will now plot and produce, they are related to passive solar-induced fluorescence. With that, first I would like to give you some background. The story goes like this. When light is absorbed by plant leaf, it can either go to photochemistry and drive photosynthesis, or it can be dissipated as heat, either basal or regulated, xanthophyll cycle that we touched upon and will touch upon further. But the third pathway is a chlorophyll fluorescence emission. The emission, as I said, happens at the level of photosystems, and here in the review, it's tried to separate effect of photosystem 1 and photosystem 2. And usually it's emitted in the red and far red ranges. As you see that to the sensor, far red fluorescence makes the most, but still the signal is very low. It's in 2.5 milliwatts per square meter per nanometer. The positions of these peaks and the height of these peaks are output in fluorescence scalars file. Now, let's go to the SCOPE model and run it. As always, we'll conduct individual runs. So in input folder, we will have to say in set options that we want individual runs, simulation 0, and that we do not want to verify anymore. So we say verify to 0. As you see, by default, calculate fluorescence, the option we discussed today, is on. Traditionally, optional change of the simulation name that I'll just call SIF. In file names file, save it and close it. In input data default, we are not changing anything. However, we go one folder backwards. In set parameter file names, we have to specify among other files that we are actually using input data default. Save it, close it, going back to SCOPE, run it. I'm going first to show you output that is available in the folder. So it's output folder, solar-induced fluorescence. Here you see a lot of outputs. The fluorescence related are apparent reflectance, then all sorts of fluorescence at leaves and canopy scale. So these 5 files. Then radiance in the observation direction, including fluorescence. And of course, sigma f, the escape probability. We output all chlorophyll fluorescence related parameters in CSV files but I'm going to use the variables because they are the same in this case, the variables that state in the workspace to plot and explain you step by step what different outputs mean. So first of all, it is very important to differentiate between fluorescence at different levels. Therefore, I would like to go back to the presentation, tell you that the fluorescence originates, as I said, at photosystem level. So light strikes photosystems and some of them emit light. These light photosystems are inside chloroplasts, inside leaf cells. So they are inside the leaf. After being emitted, this light can also be absorbed by other chloroplasts, by other cells. So this amount, as you expect, is the maximum amount emitted we can have, because while the light is passing through the leaf, reabsorption happens. So some amount of fluorescence quantiles, they are getting back to the leaf. We don't even see them. However, on leaf, we can emit fluorescence backwards, so towards the sensor, towards the observer, or forward, downward, towards the soil. So backward and forward scattering is taken care of in Fluorescence model. So we compute both upward and downward emission. And finally, at the canopy level, we only see fluorescence. Well, if we put sensor under this, we would also see photofluorescence, but normally we only see fluorescence that is emitted at the top of the canopy. So only the upward part. And this is taken care of in RTM-F. The other related parameters are, for example, in input, leaf bio FQE defines how much light, so in this case 1% of light absorbed by chlorophyll will be emitted as fluorescence by photosystem, and optipar phi defines the shape of this absorption. At leaf level, we have dependency of fluorescence on photosynthesis and stress of the leaves, which is the yield of steady-state fluorescence. So this ratio, eta, of steady-state fluorescence to the dark adapted state fluorescence dictates how much light is absorbed by each specific leaf. And in canopy, we have full profiles that we saw in the profiles lecture of sunlit and shaded leaves, which emit fluorescence differently. So first thing we're going to see is exactly these three components. The blue component with the peak in red fluorescence is what is emitted at photosystem level. This is the content of fluorescence reabsorption corrected file. While passing from the inside of the leaf to the outside of the leaf, this amount, everything that you see in blue, is absorbed. And only this, what you see in red and yellow, proceeds further in both upward and downward direction. This is hemispherically integrated fluorescence emitted by all leaves. This is the file where this output is stored. So it's the whole area. And again, fluorescence emitted backwards has to also undergo some scattering and reabsorption. And as a result, what we would measure at the top of the canopy is fluorescence hemispherically integrated, rad E out F. So only this portion is making it through the canopy, through other leaves. So you see the red portion of light is absorbed almost totally, and that's why we see far red peak at the top of the canopy as the maximum. Now, let's talk about this part that we can actually absorb. With SCOPE, we can also decompose it. In this case, I'm talking about radiances, so it's in the direction of observation. The unit of the radian is here. So this curve is what an observer would see. This is fluorescence emitted by sunlit leaves directly in the observation direction, and by shaded leaves, only this small portion, observation direction. The rest is, of course, also fluorescence emitted by leaves, but not in the observation direction. This fluorescence gets scattered within the canopy, and even this small green portion is the one that went down to the bottom, was reflected by the soil, and made it directly from the soil towards the sensor without any further reabsorption by canopy. So those are four components we can also differentiate. This output is not output, how funny it sounds. This is available in the workspace after the run, but the total fluorescence in the direction of observation is, of course, output in file fluorescence.cs3. It's a spectral quantile. If we want, and we have to, go from the fluorescence that was observed by a sensor towards the total fluorescence emitted by photosystems, so we have to go from this towards this in order to make conclusions about the state of the plant. For that, we need the escape probability, the sigma f, that is output in the file sigma f. Escape probability defines how many photons will reach the observer, but normally it's used backwards. So when we measure fluorescence, we multiply it by pi in order to represent the whole hemisphere, and then we divide it by the escape probability sigma f in order to get this curve. And this curve, this energy, tells us about the plant state. And the final output I would like to talk about is this apparent reflectance and apparent radiance. So if we exclude the influence of fluorescence, we will see reflectance and radiance presented here in the blue curve. But the fluorescence gives extra outgoing light that was not in the incoming spectrum, and that's how it's retrieved from the sensors. And this is called apparent reflectance and apparent radiance. So it's how we actually see with the instrument, how we actually observe with the instrument their reflectance. So the difference between them, between the apparent and the true reflectances and radiances, is the solar-induced chlorophyll fluorescence. Here is in percentages, so around 0.7%. And here it's in radians, watts per square meter, per micrometer, per steradian. It's exactly the same shape as this one, just not multiplied by pi. It's exactly the same shape and values as this one. If you have doubts about the photon outputs and what they mean, there is part of the documentation that describes output files, and with option calculate fluorescence, these output files will be added extra. So you can see which exactly variables are output and what they mean. That is fluorescence spectrum in observation direction, for example. And this is top of canopy hemispherically integrated fluorescence, etc. That's it with respect to solar-induced fluorescence in the SCOPE model. The topic is under development, and we are all looking forward to the FLEX mission of the European Space Agency that will provide us measurements of this quantile from space. See you in the next videos.